Waiting for recording to start. Ding, here we go. Let's talk about mutations. Let me see. Here we go. Okay, so we were talking about viruses and various ways of trying to treat those. Seems like a fairly timely topic. Other ways in which DNA can impact human welfare is through mutation. And when DNA is replicated, you might remember that the double helix unzips into two strands, right? And the two strands move apart, and then a new complementary strand is made on each old strand. We talked about that. DNA polymerases are the enzymes that actually make that happen. And if your DNA gets damaged, there's another set of enzymes that move down the double helix and repair certain types of damage. And they're really good, but they're not perfect. Every so often they make mistakes. Either the repair enzymes don't repair it correctly, or the DNA polymerases make a slip, and they can change the order of nucleotide bases. So a gene now spells something different. It would be like changing a single letter in a sentence. Sometimes if a mutation results in replacing a regular amino acid codon with a stop codon, the gene might even be silenced, no longer translated into protein at all. You can also, because of mistakes in crossing over during meiosis 1, sometimes you'll get portions of genes getting deleted or duplicated, sometimes even entire genes or even entire sections of chromosome may get duplicated or they may get deleted. All of those are what we call mutations. Any kind of error in DNA replication that gets passed on is a mutation. And so anything that can damage DNA and cause mutations is called a mutagen. There are chemicals that are mutagens. Radiation is a mutagen. If you get, too close to an atomic bomb going off or something like that, that will damage DNA. A big one that you know about is ultraviolet light, this wavelength of light that you can't see, but that reaches the ground in sunlight. All of those can damage DNA. Your body's got amazing enzymes for repairing the damage, but they're not perfect. So what will occasionally happen is the DNA in one of your cells gets damaged, doesn't get repaired right, and ends up with an altered sequence of bases from what it used to have. What you're looking at here, by the way, on the left is a little glass dish, or the edge of one, with a jello-like substance with nutrients mixed into it. It's called agar. And the little white spots that are growing on it are colonies of bacteria. Each one of those is a blob of bacteria, each descended from one single bacterium that divided into 2, 4, 8, 16, ... blob. The reason I'm showing you this is that bacterial growth can be used to check for mutations in a test called the Ames test. Basically, you grow bacteria on a medium like agar, zap them with a chemical that you think might cause mutations, and look for changes in their ability to grow. It's not perfect, but it's the first line test for mutagenic activity in new chemicals and new consumer products. So, drugs or, industrial compounds, things you might find in your deodorant or your hair dye or something like that. The first test that they'll do is an Ames test. because I don't know if you've quite twigged to this yet, but all life's got DNA. You have DNA, bacteria have DNA. There's no difference worth mentioning between the two. If we took a gene from you and stuck it in a bacterium, that bacterium would make human protein. In fact, we've been doing that since the 70s. There's no important difference between your DNA and bacterial DNA, and anything that causes bacteria to mutate can cause your DNA to mutate as well. So the Ames test is very cheap. It takes up a lot less space, a lot less resources, and maybe is a lot less, morally problematic than testing on animals. And it's the first line test for chemicals and consumer products that are released, checking for their potential to cause mutations. The reason this matters is this. We're not going to go into all of the details. There's just no time. But cells control their division with specialized proteins that you can think of as brakes and accelerators. If a cell that's okay, so there's that. Cells regulate how often they divide with some specialized proteins that either stimulate or slow down the cell cycle. The other thing is that cells that are damaged or cells that aren't needed can actually trigger this very orderly process of shutting down, turning out the lights, and putting up the chairs on the tables. It's called apoptosis, otherwise known as programmed cell death. When you were about the size of a large bean, you had little arms and legs, and they ended in what looked like little flippers. The reason why you've got separate fingers, I mean, unless you're one of those few people that have webbed fingers and toes, but the reason why you have separate fingers is because the cells that used to be part of your flipper in between your fingers and toes got a message that basically said, quietly commit suicide, and that's what they did. They activated a kind of program inside the cell that results in the cell just quietly shutting down and breaking up into tiny little blobs. And that's called apoptosis. If you've ever seen a tadpole transform into a frog, the tadpole tail breaks down because of apoptosis. The cells that hold a leaf onto a tree break down by apoptosis, causing the leaf to fall in the autumn. So this happens all the time. And that's also controlled by certain genes. You could teach a freaking course in this, and I'm giving you the tip of the iceberg. But a really cool thing that DNA can do is this. DNA includes genes that are transcribed and translated into proteins, and then the proteins can come back and bind to the DNA, and in some cases they will promote other genes, and in some cases they'll repress them. So this diagram here, the protein is shown in green. You can see lots of alpha helices there, and it's just the right shape to bind to the DNA helix. And this particular protein will block the transcription of another gene. There are other proteins that might bind to the DNA helix and encourage a gene to be transcribed. We call those promoters. So this enables one gene to act as an on-off switch for other genes. You could have one gene that's transcribed and translated into a protein that then comes around and blocks several other genes from being transcribed and translated. Or you could have a gene that's transcribed, translated into a protein that comes around and encourages several other genes to be transcribed and translated. This is part of how genes can control cellular processes like division and apoptosis. It's also part of an immensely complicated field we just haven't time to get into, and that's the field of development. How you go from a single fertilized egg to a full adult phenotype. How does that one fertilized egg know how to, as it divides, transform into muscle cells and bone cells and brain cells and all other 200 types? A full answer for that, would take a lifetime. We still don't have a really full answer for that, but the outlines of how that works are quite clear. And a big part of it is that genes can act to control other genes. All of the cells in your body, most of the cells in your body contain all of the DNA that the original fertilized egg had. It's just that the genes that each cell isn't using are inactivated. So your liver cells still contain the DNA needed for making brain proteins, but that DNA has been shut off, and the only DNA that's actively transcribed in liver cells is the genes for making the proteins that the liver needs. And this is part of the story too, is that genes can block other genes and shut down other genes, but also start up other genes. And that's the tiniest little taste of how your cells can actually do this sort of thing. Genes control other genes. Here's the thing. If the control genes, those accelerators and brakes, are mutated and they don't work as they should, the cell may start dividing uncontrollably. So just like a car with a stuck accelerator and no brakes will just start speeding down the hill, a cell with damaged control genes, called oncogenes, by the way, will start dividing and dividing and dividing and not be able to stop. That apoptosis thing that I talked about is kind of the fail-safe, in that a cell that starts dividing uncontrollably is supposed to switch over to apoptosis and quietly break up before it can do any damage. But cells with mutations often lose the ability to undergo apoptosis. They lose that self-destruct switch. So they'll divide, they'll divide, they'll divide, they'll divide, they'll divide, and they can't stop. The result of that is cancer. So anything that damages DNA is a potential cancer-causing agent. Anything that's a mutagen, which causes mutations, is potentially a carcinogen, a cancer-causing substance. That's how UV light can cause skin cancer if you spend too much time at the beach without sunscreen, or too much time in the tanning bed, or something like that. That is how certain chemical compounds, exposure to them, like asbestos, for example, can give you an increased risk of certain types of cancer. Those chemicals are the sorts of things that damage DNA. This is also why some types of cancer tend to run in families. There's a gene called BRCA1, and if you inherit a particular allele of BRCA1, you inherit a gene that's no longer as effective at controlling cell division, and that increases your risk of breast cancer. So you can now get tested for your BRCA1 alleles. Some women who test positive for the dangerous alleles of BRCA1 actually choose to have mastectomies before they even have cancer, because their risk is so high that they're afraid that they're going to catch it down the line. I don't know that that's always justified, but certainly it's something that you have to watch out for if you've got a family history. This is why some types of cancer do tend to run in families, if you inherit an oncogene that's already not working right. Not all mutations, however, lead to cancer. Probably most mutations actually don't do anything. Some mutations will change one codon to another codon for the same amino acid and have no effect at all. Some mutations will change the primary structure but don't affect the shape of a protein. Some would change the shape of a protein but not an important part. Just like changing the shape of a screwdriver handle may not really affect how useful the screwdriver is. Some, of course, do change the primary structure, and that leads to major changes in protein shape. Some might even stop a protein from being made in the first place. Are they ever beneficial? The answer is you can't answer the question without looking at the context. You can't answer by looking at the environment. Probably most mutations have no effect at all. It's been worked out that every human is carrying something between 1 and 10 mutations. You're carrying alleles that were not present in either your mom or your dad. There are some genetic diseases that can pop up in a family with no history of them because of mutations in a particular gene. So we're all mutants to some extent, and the vast majority don't do anything detectable. We call these neutral mutations. Some are pretty much always harmful. Others may be beneficial depending on context. And bacteria have been used a lot to look at this. A bacterium called Escherichia coli will divide 6 times a day, which means you can look at change over tens of thousands of generations. And a team led by Dr. Richard Lenski at Michigan State has been growing the bacterium Escherichia coli in culture ever since 2001. So his team has observed change over 70,000 generations. That's a lot. The results seem to be that roughly 1 in 12 mutations actually cause benefits to the bacteria. They give them increased efficiency in using the food that he gives them. In fact, in one well-documented case, there was one set of mutations that gave the bacteria the ability to use a food source that they couldn't before. They were able to use a food source called citrate that previously they could not. And he was able to pinpoint when the mutations happened and exactly what they did. Some really great work that Dr. Lenski has been doing. And it can happen in the wild as well. Those are some bacteria called flavobacterium because they've got big clocks hanging around their necks. Actually, I'm kidding. That would be flavobacterium. These particular ones were living in wastewater ponds at a factory that made nylon. They were discovered in 1975 in Japan. I'm not sure if they were ever given a formal scientific name. They're usually just called Flavobacterium K172. And these bacteria were able to break down byproducts of nylon manufacture. They were able to break down waste products of nylon. The reason that's cool is that these nylon did not exist before 1935. It's an artificial fiber. And so we know that the enzymes that K172 uses came into existence sometime between 1935 and 1975. And it's been possible to track the mutations in existing genes that gave Flavobacterium the ability to break down this food source that nobody else could use, right? Think of natural selection, right? If you're a bunch of bacteria in a pond, you may be competing for food sources. But if you can use some chemical that nobody else can, then you have it all to yourself, and you can, make use of that resource and make lots more bacteria that are just like you. So selection has favored the evolution of bacteria that can break down chemicals that, as far as we know, did not exist on the planet before 1935. So that was a case of a mutation that was very beneficial for the bacteria because it lets them use a resource that no other bacteria can. So heck with bacteria. What about people? There's a condition that people have called Marfan syndrome. And if you have it, you have a gene for an altered form of a protein called fibrillin. Fibrillin's found in the extracellular matrix. The Marfan's allele is dominant to the normal allele, so you only need one copy. You need one dominant and one recessive to produce the syndrome. So it can run in families, but somewhere between 15 and 30% of all cases are caused by spontaneous mutations in the fibrillin gene. So it can pop up in people that have no family history of it. Paganini, by the way, possibly the greatest violinist of all time, is suspected to have had Marfan syndrome because it tends to make you very tall. It tends to give you very long fingers and toes, and it makes your joints very mobile. So very tall, very long fingers, very mobile fingers, very useful for a great classical violinist, one of the best ever. We don't know because he died before testing was possible, but he's suspected of having had Marfan syndrome. Oh, this is another guy with Marfan syndrome, a Spanish actor named Javier Botet. And yeah, you can tell he's got a very tall and slender build. And he's actually able to use this to great effect because he's one of the most sought after actors in horror movies because he's naturally very gifted at playing monsters that are very tall, slender, and flexible. There was a movie a few years ago called Slender Man. I don't know if any of you saw it, but he played the Slender Man, of course, in very heavy makeup. You know, the Slender Man doesn't have a face. He was in a recent remake of the movie The Mummy. He's been several monsters that are tall and skinny and scary, and he's carved out quite a career because he's physically very well suited to play these roles. All of this results from the body's making an altered form of the protein fibrillin in its connective tissue, part of the extracellular matrix. And so you can see the Marfan phenotype. They tend to be very lean. They tend to be really tall. They've got very long arms and legs. And they tend to have very loose joints. They are, as we say, double jointed or hypermobile in their hands and feet. And here's the thing, they would be naturals at sports that require reaching and stretching and height. People with Marfans would be absolutely killer basketball players because they tend to be really tall and also have really wide wingspans, right? And have really big hands, just the kind of thing you would want for, swatting away a Kareem Abdul-Jabbar skyhook or something like that. They'd make very good volleyball players with their height and their reach and their big hands and their big span, except for one thing. Marfan syndrome also leads to weaknesses in the walls of major blood vessels, notably the main artery carrying blood away from the heart, the aorta. Here you've got a partially dissected heart, and you can see there's a bulge at the root of the aorta, like the bulge in the sidewall of a tire. And the problem with bulges in your tires and bulges in your aorta is that they'll burst without warning. And an aorta that ruptures is usually very fatal very quickly, and it can happen with no warning. You can die, you can collapse and be dead before the paramedics even get there if your aorta ruptures. And so people with Marfans can live perfectly healthy and productive lives, but vigorous sports are very risky. Anything that puts strain on your heart, makes your heart pump really hard, is a risk for making those aortic bulges, the aortic aneurysms, for making them worse or even making them rupture. And people with Marfan's get heart scans very regularly and may go in for surgery if an aneurysm is found, because you can repair these surgically today. The problem is you may not know that you have them until it's too late. So this was a college basketball player in '76. You can tell that's an old photo because the shorts are so short. Chris Patton at University of Maryland had a great freshman career, was playing a pickup game in the summer of '76. He went up for a jump shot and was dead when he hit the ground. His aorta had spontaneously ruptured. He had Marfan's, but it had never been diagnosed. Same for Flo Hyman, great volleyball player who led the U.S. Olympic team to a silver medal in 1984. One of the best we've ever had playing the game, went on to play professionally in Japan and rotated out of the game, sat down, and then never got up again. She died of a ruptured aorta in 1986. There was another woman in 2007 who went to sleep after a hard game and never woke up again. Ronalda Pierce. She also had Marfan's syndrome. So if you have it, you can lead a perfectly good life, but if it's not diagnosed, it can give rise to these weak spots in your aorta, and those can kill you suddenly without any warning. So people who have Marfan's are these days encouraged to take up very low impact exercise like golf or walking or things like that. And of course, they get their hearts monitored very, very carefully because you can fix these with surgery if you catch them in time. So that's a mutation that can improve your athletic abilities, but can also be really bad for you because it increases your odds of sudden death from aortic rupture. Are there mutations that are completely beneficial? This is a guy from the country of Finland at the 1964 Winter Olympics in Innsbruck, Austria. His name is Eero Montiranta, and he was a champion cross-country skier, winning 2 golds and a silver, blowing away the competition in his gold medal races. I think he came in like a couple of minutes ahead of his nearest competitors. He was just insanely good, one of the best endurance athletes ever. And it turned out that he had a mutation in a gene called EPOR. It's called erythropoietin receptor. It enables certain cells to respond to a hormone called erythropoietin. So it's another one of those receptor proteins like ACE2 that we saw in the last lecture. And that mutation made him make extra red blood cells in his blood. And it turned out that at least 5 generations of his family had this trait as well. This is part of his pedigree chart. Hopefully you remember how to do pedigree charts after doing Lab 5. Montiranta himself is at number 17, and you can see there's an arrow pointing to that square that filled in. The fact that it's filled in means that Montiranta had this trait of making extra red blood cells, right? Number 18 is his wife. She's a circle because she's female, and she's not filled in because she doesn't have extra red blood cells. And then you can see he passed it on to one of his daughters. He had a daughter and then twins, a boy and a girl, and the girl had the trait because she's shaded in. You can see two of his sisters had it, 15 and 19. Their father, number 3, had it. There's a slash through him because he was dead when this study was done, and so on. And it turns out to be inherited as a plain old Mendelian dominant allele. So Montiranta is probably heterozygous for it because some of his kids didn't get it and others did. And the same is true for Montiranta's father. This follows the pattern of a simple Mendelian dominant allele. And it goes back at least 5 generations. They're not sure when it began, but they've been able to trace it that far. This is him and his family, by the way. He is sitting down in front. He and his wife are up there in front. You might be able to tell he's got kind of a red complexion because he's got extra red blood cells that actually makes his face look ruddy. And you can pick out the people in this picture who have this trait because they tend to be kind of red-faced. Now, some people who make extra red blood cells are at elevated risk of stroke or other blood clotting disorders. But for some reason, Montiranta's family didn't suffer any bad effects. They lived just as long as their kin that didn't have this condition. They were perfectly healthy, except that their blood could transport 50% more oxygen than a typical person's blood. In other words, Eero Montiranta did naturally what some athletes try to achieve with something called blood doping. You might have heard of athletes getting blood removed from their bodies, their red blood cells get stored, and then injected back into them on game day gives them the ability to transport more oxygen for their muscles. This is illegal, by the way. This will get you kicked off the team if you're caught. But his body did that all the time. He just naturally had a bit less than double the amount of red blood cells you'd find in a normal person with none of the side effects. And the result was that he was one of the great cross-country skiers of all time. Cross-country skiing is, there's people that buy expensive machines to duplicate what cross-country skiing does to you, right? You've seen Nordic track machines and things like that at the gym. It's a heavy workout for both your arms and your legs. It's one of the most physically demanding athletic events that's out there, probably more so than marathon running. And Montiranta got his amazing endurance because he was carrying this mutant allele that gave him an extra helping of red blood cells in his blood, which meant that he could transport that much more oxygen to his muscles. So most mutants do not end up like Nightcrawler or Cyclops or, the rest of the X-Men. But every so often you do get a human mutation that gives you maybe not unbelievable superpowers, but certainly superpowers compared to the rest of us. So there. There's one more gene I'll talk about that's associated with athletic performance. This is a computer model of a protein called alpha-actinin 3. It's made up of 2 peptides, each of which has multiple alpha helices. You should be able to recognize this stuff by now. And it's involved in muscle function. This is a muscle cell that has been stained with a fluorescent stain. The black area is where the nucleus is. That doesn't contain any muscle protein. Almost all of this muscle cell is packed full of proteins that are involved in contraction. And in this photo, ACTN3 has been stained fluorescent red, and you can see that it exists in these narrow stripes in a muscle cell. So that's where it's located. And it turns out that alpha-actinin 3 exists in several alleles. One of them appeared roughly 50,000 years ago, and it's estimated that about 50% of Europeans carry this mutant allele. Roughly 18% of Europeans are homozygous for the mutant allele. The mutant allele is a bit more common in Asian populations and a bit less common in Africans, but it's certainly present in all human populations that have been studied so far. People of any race can have either normal alpha-actinin or mutant alpha-actinin. The mutation is called R577X. Okay, so what? That mutation is a knockout mutation. If you have it, you don't produce the functional protein. You don't make alpha-actinin 3. For most of the proteins in muscle, this would be really, really bad. There's something like 50 different proteins involved in muscle contraction. And in most cases, if you don't make one of them right, you end up with a form of muscular dystrophy. Meaning muscles losing their strength, ending up in a wheelchair, unable to move on your own as your muscles lose their function. Alpha-actinin 3, however, if you don't have functioning alpha-actinin 3, there's a different protein that can step up and take over called alpha-actinin 2. So you may not even know whether or not you can make alpha-actinin 3. And for the vast majority of us, it makes no difference at all. If you get your alpha-actinin 3 knocked out, you can simply replace it with alpha-actinin 2. However, back in 2003, they did a genetic study of Olympic sprinters and showed that every one of them were at least heterozygous for normal alpha-actinin 3, and none of them were homozygous for the mutant allele. The same pattern was found in weightlifters, who are like sprinters in that they have to generate lots of power fast, right? If you're, picking up an enormous barbell, right, you can't, you've got to be able to generate this huge surge of power in order to be able to, snatch and jerk it up over your head. Sprinters, it's the same thing. You have to generate huge amounts of power very fast in order to explode off the blocks and get to the finish line. And it seems that having the normal alpha-actinin 3 gives a slight advantage in speed, in being able to generate muscle power quickly. The mutant allele turns out to be more common in endurance athletes, more common than you would expect by chance. It seems that having that mutant allele causes your muscles to use energy just a little bit more efficiently, and that improves your endurance ability. You are just a little bit more efficient at using that energy, and that improves your ability to exert power for long periods of time. They found the pattern in a 2012 study of elite male athletes, elite endurance athletes, meaning, marathoners that can challenge for, national titles and things like that, not just people that run them for fun, but, people that train very seriously, were almost twice as likely to be homozygous for the mutant allele as elite power athletes like sprinters. For world-class endurance athletes, they were almost 4 times as likely to be homozygous as national class athletes. This gene by itself does not guarantee that you're going to become a champion marathon runner, but it increases your chances of becoming one if you train. Because it gives you that slight little edge in efficiency. And in fact, when this was discovered, some company developed genetic tests that you could give your kid that would tell you whether your toddler is predisposed towards endurance sports like long distance running or cross country skiing, or power sports like sprinting and weightlifting. So you can start trying to line up college scholarships for your toddler by figuring out what your toddler is most interested in and steering them towards those sports. Not interested in, who cares about that? But steer your toddler towards sports that he or she may be most physically, genetically gifted in. How useful this is, probably not, but somebody's making money off of selling genetic tests like this to a certain class of moms. So you can find out whether you or your child is a natural for endurance sports or power sports. The thing is that ACTN3 is only one of a lot of proteins that are critical for muscle function. All of those proteins have to work together. And what you've got is a lot of epistasis. Remember, that's what you have when you have multiple genes working together to affect a particular trait. In fact, they've identified at least 200 genes that seem to have something to do with your sports performance. Genes that affect your body's ability to transport oxygen, heart function, nervous system. Lots of genes play roles in athletic performance. There is no single gene for being a great athlete. Someone once said that anybody who wants to win an Olympic gold medal should select his parents very carefully, because so many genes have to be just right for you to be an elite athlete. And then, of course, you still have to train. All right, there is no gene that will turn a couch potato into an Olympic medalist. So ACTN3 doesn't make any difference to most of us because most of us are not trying to be champion sprinters. It won't make the difference between a couch potato and an Olympic athlete, but the idea is that it might make the tiny difference between a sprinter who wins Olympic gold and a sprinter who comes in fourth. It makes the difference between, a world champion and a really good athlete who doesn't manage to win any medals, right? That tiny fraction of a second might be all that separates a really good athlete from a superstar, and ACTN3 might just be enough to make that very small difference, right? Most of us, I don't know what alleles of ACTN3 I've got, and I don't care, but, it might make a difference if I was trying to be an Olympic medalist. But even then, this doesn't work for most sports. There was a study of judo athletes, judoka, that showed that there's no association between your ACTN3 alleles and elite rank in judo, which is not really either a power or an endurance sport, right? You're not trying to generate force to flatten your opponent. You're trying to use your opponent's strength against him. So it doesn't really count as either type of sport. And it doesn't matter what your ACTN3 alleles are. They don't have any effect on your chance of becoming an elite judoka. And there's also no association between those alleles and being an elite player of soccer, basketball, or hockey, probably because those call for such a mixture of skills and abilities that there's no single allele that gives you an edge. Right? Soccer players have to sometimes make fast breaks and run quickly. They've also got to endure long games. And some players might be a little better at one or a little better at the other. So maybe there are some players that are great for making breaks, but they have to rotate out sooner or something like that. But there's no single allele that gives you an edge because you have to do several different things. And you have several different, abilities that you have to call on if you're playing soccer or basketball or something like that. So mutations, sometimes beneficial, sometimes harmful. Often it kind of depends because you have to take into account things like epistasis when you're trying to understand the effect that mutations have. Right. I got 1051. We'll come back at 11:05, and I've got just a little bit more to flap my gums at. And I will go ahead and stop the recording now.